From MicrobeTV, this is Office Hours for Wednesday, January 28th, 2026. I'm Vincent Racaniello, and you're not anything. Welcome to the viral corner of the internet. I was in podcast mode. You're listening to the podcast all about viruses, but this is not a podcast. This is a live stream. Welcome, everyone. Welcome to our moderators. Who do we have tonight? Les Fabi is here. Tom Steinberg is here. Vanity Nutrition is here. Thank you for coming. It is like -9 Celsius tonight in New Jersey. It has been frigid. We got a foot of snow. It's still hanging around. They can't put it anywhere, right? It's all stacked up on the streets of New York. You're lucky if you can cross the street. But hey, we forge on. Today was lecture number 3 in my virology course, Genomes and Genetics. But tonight, what do we have for you? But first, welcome, everyone. I'm glad to see you here, soaking up some knowledge in the form of virology. Please let us know where you're from so I can shout you out. What do we have tonight? Always jam-packed stuff. Besides you, of course. You're very important. I'm going to talk about Dr. Kirk Milhone, the new chairman of the ASIP, the Advisory Committee for Immunization Practices. This man should scare all of you. We have a mini lecture, and then to wrap it up, some poetry. Let's see who's here. Rita is here from Idaho, where the day is ending clear and 4C, though it started with blowing snow. Ooh. Here's the virology course link. 2 lectures are up so far, and the whole 25 will be posted to a week. Dagfinn, hello. -10 and 2 a.m. in Oslo, Norway. Still outraged with Agent Orange, who claims to have sent a beautiful armada towards Iran. There's nothing beautiful in military activity. The man is an asshole, as is everyone else in his administration. Their actions show just lack of humanity on all sides, and I can't wait for them to be gone forever. Unbelievable. Hello, JJ. First-time listener from Florida. 10C feels like 5C. Pretty cold for Florida. Yeah. JJ has a question listening to TWIV 1291. The question is, do some children who get NOPV2 develop polio paralysis, or is it only unvaccinated children? That's a very good question. So certainly it could, right? But whether it does is not clear. When I first started following the use of NOPV2, I understood that it had caused paralysis in a number of children somewhere in Africa where it was being used. Not Uganda, but maybe Burundi and a few other places. But now, a paper that we discussed on TWIV 1291, they say no recipients got paralysis. So I think that's not a complete story. I think what's happening in other countries needs to be looked at. So yes, it could. I thought it did, but we'll see. MN, chilly in DC. I'm sure it is. Tom is in the Oregon Coast Range. 0F, Claudia was in St. Paul, Minnesota. Got home last night. I think you're brave. Maybe that's the other side of the river, right? The other part of the Twin City. Kip and Laura, thank you for your support of science communication. Right now, Laura and I are at a candlelight vigil at her med center in honor of those killed by ice. Catch you all later. I don't know how anybody can support just killing people who are are innocent. I don't know how you can support it. Even people who still support the Orange Man say it's fine. I don't know how you can do that. You're a gross human being if you support that. Simply gross. And the thing is, scientists and public health people and doctors work hard to make medicines and vaccine to protect you. And you show no great gratitude whatsoever. MN asks, do services like 23andMe measure Pandora virus? No, they don't not doing diagnostic tests like that. They are just 23andMe and probably the others too. They're just looking for single nucleotide polymorphisms, that is, single base changes in your genome that are associated with various conditions, right? Like color blindness or earwax or something like that, and many other things as well, but no infectious agents, no. Will is in gloomy Changsha where it's 5 degrees Celsius. Why is it gloomy? Because of the weather? I guess so. Carol, our favorite nephrologist, Bakersfield, California, 16 Celsius. Yes, 16 is much better than -9. In fact, overnight it's like -15. Now, I know there's some places in the world that are much colder. I understand that. But New York rarely gets that cold. I can't remember when it was that cold. John is in Minneapolis, -15. There you go, even colder. Trying to quiet the orthogonal brainwaves with some virology tonight. It's very disturbing that nobody owns up. No one shows any compassion. Nobody tells the truth. Everybody's a frickin' liar in this administration. To a single person. You show me a person who isn't a frickin' liar. Michael Schmidt. Hello, Michael. Should we be concerned with the case of Nipah virus that has been detected in India? Should the U.S. and the globe be concerned? No, we should not be concerned. Nipah virus, first of all, the infections occur in areas where flying foxes are present, and that's Southeast Asia, Australia. It's a wide area, but it's not here. But you could argue that maybe someone could get infected and travel. It doesn't transmit very well from person to person. You have to really immerse yourself in an infected person. What do I mean by that? I mean that you have to. So, for example, in many countries, the family prepares the dead for burial. So you're really getting close to the body fluids, and that's how you get transmission. But casual community transmission doesn't occur. And so there have been a number of cases in parts of India and Bangladesh, but haven't spread, which doesn't mean it might not, but I'm not worried about it. I don't think it's high on the list of concern at the moment. I'm concerned for people who get it, but not for that spreading. Mark Owen Martin, 8 degrees in Tacoma. Haven't seen any snow, just a little frost in the morning and ice on my little koi pond. So the koi are okay? I guess it doesn't get that cold there. Yeah, we have a lot of snow here, Mark Owen Martin. Douglas is in Redondo Beach, chill, 64F. Redondo Beach. Vanity Nutrition, it's a brisk -7 on Long Island. Yeah. Kang is in Chicago where it's -10C. The sun was out this afternoon. The sky will remain clear tonight. I don't know what it's doing outside the sky. I didn't look up as I was making my way home. I didn't know I'd make it in time because New Jersey Transit's screwy. My train was 10 minutes late departing from Newark, but I made it in time. Peter, hello. Haven't been here in a while. Hope everyone's been okay. Well, there are lots of problems in the U.S. Peter, and so we're not okay. We're very much disturbed at the state of matters in this lying country where people can no longer tell the truth. I just don't get it anymore. People shouldn't vote for any of these people because they're liars. Hello, Questella. Super cold here still. Indiana, I guess. Gallbladders in Franklin, Tennessee, 23F and ice everywhere. So that's freezing because 32 would be freeze point, right? Helen from Western Massachusetts. We got 34 inches of snow last weekend. It's clear at -13. Holy cow. I guess Alan Dove got 34 inches of snow. I'll have to listen to him on Twiv telling us about how he cleared it. Is my audio volume lower than usual? No, it's right here on the USB, on the the volume meters. It's going up. I could crank it some more. Let's see. Here's some more. That's now going into the yellow if you want. I had to change my interface because the one I was using died on Monday. I was recording Beyond the Noise, or I was about to record Beyond the Noise. I couldn't get it to work, so I had to wear headphones. It was a pain. And I have another one here. It's okay. It's fine. But my old interface, I combined audio and video. It was much better. So, and you may see there's some delay here, a little bit of delay when I talk, and you see my lips move. If there is, let me know. I can add more of a delay to try and neutralize it. Anyway, is the audio okay now? Let's see. All right, what else do we have here? Sandra with a new icon, 16C from Mexico. Welcome. Abdul Rahman is in Brooklyn. Welcome, Abdur. Heather is in London. Cool. It's probably warmer than it is here. Lagotic is in Chicago, -11C. Luther is in Rio de Janeiro. Very nice. It must be summer down there, right? Elizabeth is in a very cold West Virginia. Betty is in very cold Washington Heights, 18F. Jeff is in Elgin, Illinois, 12F and clear. Here we go for London, 4 degrees Celsius. Pretty cold, but not freezing. Peter, nice to have you. Tona, hello from a cheap, it's not cheap, crisp, fresh, cold air, Long Island. I'm hearing an echo in my audio, so I'm going to have to do some more troubleshooting. Oh, well. Audio better now. Okay, good. This delay, this is, the problem is the previous interface I had, which died. You could put the video and the audio in it so they could travel together to the computer. Now here I have a box for video and a box for audio, so there's an inherent difference in the time it gets for the video versus the audio. So video is really fast. Audio is slower. Anyway, Mike from Seattle, where it's 9C and cloudy with light rain. Maybe it's my other headphone. I have the other one out because... So I usually have 2 earbuds here. And the other one I took out. You can see it's just hanging here because it has a delay. My left ear has a delay, but the right ear doesn't. I don't really need to hear myself because there's nobody with me talking to me, but I like to hear myself. Abdul Aziz is from KSA, 15C. That's a respectable temperature. Yes, Sandra changed her picture, a smiley picture with, is that a cat there? It's very nice. Can we get some comment on the Nipah outbreak in India? I did, right? Did you hear me talk about that in response to Michael Schmidt's question? Maybe you posted that before that. Okay, let's see how many cases there are. Okay. 2 cases. And all the identified contacts have been quarantined and tested. 196 contacts still tested negative. The cases were 2 nurses at a private hospital in West Bengal. One of the nurses is in critical condition. I presume the other one is not, because they're not saying anything here. Of course, Nipah virus is a paramyxovirus, same family as measles virus, but far deadlier. It's an enveloped RNA virus with a negative strand genome with glycoproteins in its envelope, and we have made actually a vaccine, a glycoprotein spike-based vaccine, which is being tested, I believe. John says, stop me if you've heard all this, but I thought susceptible rhymes with receptacle and permissive rhymes with replicative. That's fine. Susceptible, receptacle. Yeah, receptor, susceptible, and permissive rhymes with replicative. I don't think permissive rhymes with replicative at all, but if you think it does, that's okay. Zupski is in the Shar Mountains of Kosovo, where it's 1C. Welcome. Nice to have you. You know, some people are saying the sound is low, but it is as high as I can go. You know, that's this interface. It's just not as powerful as the other one. So let's see. I'm going through Audio Hijack to put a delay on here. Let's see if that's doing anything. Yeah, let's see. Oh, look, left channel default, right channel default. I guess I could fix the channels. Yeah, I could. So I could fix my earbud thing, but I don't want to do it now. Output volume 100%. I'll work on it offline, not now. Let's see. Stephen from Texas, welcome. Should we fear Nipah? No, as I just said. Sound is just right. Thank you very much. Ubalubi from Little Rock, 1C. Been trapped inside since Friday night due to road conditions. They don't clear things here. Not safe to venture. I guess you have to drive to get anywhere, right? Huh. Case Bainbridge. What's the latest research on cat-to-human infection of toxo and links to bipolar and or schizophrenia? As far as I know, there are none. We have talked to experts on TWIP who have said there is no connection between any neurological issues in humans and toxo infection. But you know, everything is fluid. Everything can change. So, you know, it was a few years ago, although we could ask Daniel on TWIP, which, if I'm not mistaken, is TWIP Friday? No, it's not. It's next Friday. But I can ask Daniel. Let's see, toxoplasma and neuro disease. Neurological effects. Because toxo can make cysts in your brain, right? So that can disturb transmission and so forth. So here's a paper from 2021. Neurological and neurobehavioral disorders associated with Toxoplasma gondii infection in humans. So this parasite infects 30% of the world's population. Although most latent infections in humans have been considered asymptomatic, studies in rodents suggest phenotypic neurological changes are possible. So several studies have therefore examined the link between T. gondii infection and diseases such as schizophrenia, epilepsy, depression, bipolar, dysphoria, Alzheimer's, Parkinson's, and OCD. To date, there is varying evidence of the relationship of T. gondii to these human neurological or neurobehavioral disorders. We found that schizophrenia was most frequently linked to T. gondii infection, while sleep disruption showed no linkage, and other conditions have mixed support. However, infection as a cause of human neurobehavioral disease has yet to be firmly established. So these are associations, right? They're observational studies. So you may see some association of T. gondii to of schizophrenia, but it doesn't prove that it's there. Okay, so you need to do much more. You have to do bigger studies and get more confidence that it's happening. Thank you for asking. Rainy eastern North Carolina, damned cold, but nothing like days in Chicago. Razor, hi everyone, it's 59 here in NorCal, but feels colder. Maybe the flu, which started for me this morning. It's a real bear, this one. Got vaxxed in early November, but got me some office hours. I hope you feel better. Do you want to take Baloxavir or Tamiflu? You know, you can get a coupon for, it'll be like $50. John is in Pratropkirikon, 21C, where there hasn't been any snow since the last ice age. Pratropkirikon. I want to see where that is. Let's see. Where is it? Western Thailand. It looks lovely. Yeah, I can see where there's no Pratropkirikon. Yeah, on the west coast of Thailand. Look at that. Oh, that little peninsula that sticks out below. It's kind of looks like Baja California. It's on the top third of that. Nice. It must be lovely there. Never been to Thailand. Starman. Hello, Professor. Got an awesome semester this year. Got to work with bacteriophage lysate and optimize it for DNA extraction. Sounds like fun. There's nothing better than working with viruses. Enjoy it. Have a good semester. Pete has a TB question. Well, I'm not a TB person. I've been digging into ancestry on my great-grandfather. I found a 1891 court document from Stafford, Assise, where he had a 4-month a 4-month prison sentence for some sort of fraud. He died of TB in 1895. Is prison a good place to catch TB? Yeah. Is it quick enough to kill in 4 years? Yes. A lot of people had TB back then. Let's look it up, though, how long it would take to die of TB. Killing time. No. I want to know how long it would take to kill a human, not to itself. Within 5 to 10 years of the disease developing, 1/3 might die within 1 year. Others could go longer. It really depends on you, right? Your constitution. And there's more to this, I believe. Okay, let's go back. Yeah, I said last time it was not going to be easy to arrive on time. I got to Penn Station at 10 before 6, and my 6:03 it said delayed. So I got a 6:01, which I don't usually take, but it was very slow, and I thought I would miss my connection. But my connection was delayed 10 minutes, so I made it. I got here at 7:20, had a quick dinner, and I'm here. But, you know, there were 22 more lectures left in this virology course. Plenty of time for the trains to screw up. So if I'm not here at the 8 o'clock, just sit tight. I'll be here. Environmental. Sandy, minus 17C here. Lots of snow in the western New York wilds. 85 seconds to midnight, and nobody seems to care. Huh. I don't know what that means. Jefferson is in Miami, where his concrete home, or her concrete home, their concrete home, is a freezer tonight. Oh, I'm so sorry. Patricia's in Massachusetts, cold with 2 feet of snow. Jeff writes, it must be understood that the GOP has replaced our Constitution with their unitary executive. This happened when Doge unconstitutionally and thus illegally started firing federal employees. I mean, that's been found. Judges have ruled against that, but I don't understand why nothing can be done, right? I just don't get it. Follow the Reality wants to know why I fall off at Angela Rasmussen and Peter Hotez are so eager to discredit vaccine critics with an audience. It's almost as if there's money involved. There's no money involved with me, as far as I know, and Paul and Angie, and I don't know about Peter. Now, why do we do? Because people say, did you hear what so-and-so said? So we have to say, okay, so-and-so said this, and this is why it's BS. So that's it. Very simple. Tom, 50F in Deadwood, not 0F. Saturday night, St. Paul Vigil, 8-8. That's 8 below F. Citizens are outraged. Good. They should be outraged. They should be outraged all over the U.S. at what the crap is going on. Indefensible crap with no remorse. None whatsoever. I mean, why do you beat someone to the ground and then shoot them in the back? What kind of, even the freaking police wouldn't do that. Well, maybe they would. Military, yeah, maybe they would too. Doesn't matter. It's bad. It's wrong. Okie dokie. Sandy, climate change, overshoot, PFAS, microplastic, it's all being swept away by a wave of denial as bad as sea level rise. Do I know if Dr. Offa has conflicting interests? No, I don't know. I don't ask him that stuff. Yeah, I mean, what does he have to do with? I don't know. I just don't know. I'm sorry. Carol says, I not only learned about microbes on your podcast, this week I learned what a wobble is. Who knew? I don't know what a wobble is. A wobble is referred to as the fact that some amino acids can be encoded by more than one amino acid, more than one codon. So, it basically refers to the third base of the codon that first pairs with the first base of the tRNA. This pairing allows one tRNA to recognize multiple codons. So that was called the Wobble base because it can pair with so one tRNA can pair with multiple codons. That's the reason it's called that. That's the Wobble. Mark says, I use my middle name because there are a lot of Mark Martins out there, not just a retired NASCAR driver, even a cell biologist. Gadzooks is in Vancouver, where it's 48F. Yeah, so Costella is celebrating. Is it your birthday? Is it your kid's birthday? Let us in. Hello, Joel from Bath. 4 degrees because it's late. Yeah, I heard there might be a nor'easter this weekend. Yep. Oobalubi. Tonight I made what I like to call it all soup. It has pork loin, chickpea, broccoli, garlic, all the spices, quinoa, tomatoes, bell, chipotle pepper, and Greek yogurt. No relatives in it. Larry's enjoying their video from Vancouver. Very Nika. Privyet. Hey, everyone from Moscow, where we're totally snowed in and minus 12°C, going to get colder. And back from a lovely trip to St. Petersburg. Hope everyone is okay. All right, I'm sure you had a lovely trip and the weather was lovely, but it's in Florida and it's a horrible state. The people could be good people there, but, you know, they have silly health-related policies. Good evening from Northern Maine, IFS. Very reasonable 3F. The nor'easter is predicted to mostly go offshore. Eastern Mass should get a wind, a bit of snow. Nantucket might be buried, and maybe Long Island. Well, I think Oh, St. Petersburg, Russia. Sorry. Of course. I'm very sorry. I'm stupid. I mean, I thought here that Veronica is flying off to Florida, but no. Yeah, I heard St. Pete is very interesting. I wish they would let Americans in again. Maybe one day I'd love to visit. But right now, forget it. Sorry. Yeah, St. Petersburg, Russia. Sorry. Sandy, I'm definitely going to try to visit the incubator this summer before my brother sells his house in Irvington and moves to the Cape. Irvington. I know where that is. I've been. It's not too far. Not too far. Now I can't unsee the lag between my lips and the sound. Yeah. I can put a delay in. Let me do that right now. I think I can do it live. Here, we'll go to preferences. Right now, I don't have a delay. So let me increase it to 7 or 8. 8. Check. It's still a delay. Let's go to like 30 frames. That's too much. 16 frames. It's all for Cocta. I ain't going to fix it here. I'm sorry, Logotic, that you can't see it. Pat Campbell is in Nova Scotia, minus 10C. I use Ecamm Live, yes. It's fine. It's the same camera that I always use. It's just the interface. You know, you need an interface to plug your camera and your mic into the computer, which sends it to the interwebs. And my interface died, that's all. But it'll be fine. It'll be fine. Bats. Yeah, flying foxes eat date palm sap, leaving their virus-laden saliva and urine mixed in. Starby in Chicago, minus 12 with feels of minus 15. I work the grave shift outside sometimes, and it can be rough. The graveyard shift. Polka Dot Pup is in Texoma, 0C and 8 inches of ice and snow on the ground. Gates are all frozen in, and I'm trapped. Yikes. Heard the bad news from Amy on Twiv, prompting me to ask if doctors have a legal duty of care and can be sued. I've asked that of Paul Offit and Daniel Griffin, who are both doctors, and they don't know. Right? Those are the guy, that article was all about saying, you know, hepatitis and polio are the price to pay for medical freedom. Another idiot. You can have freedoms. You can decide to take or not to take a variety of things. But if they're going to help you, why would you decide against it just on the basis of freedom? It doesn't make any freaking sense. Anyway, it's a good question, Kang. I don't know the answer. Sound is good. Don't worry about it. Okay. Thank you very much. I've noticed that some people have issues with the sound and others don't. I regularly get emails from people. Your sound has degraded in the last year. It's horrible. I can't listen anymore. So what is it? It's not degraded. It's not horrible. I listen to my product. Are you just looking for attention? Oh my gosh. Now here, if you say you can't hear me, that's fine. But you email me saying your product has degraded audio in the last year? Come on. What the hell? Let's talk about science. Like this. Well, how reliable is PrEP for preventing HIV? I have the numbers on that. I can look them up right now. It's in my HIV lecture. Let's look it up. Pre-exposure prophylaxis. Let's go to the 2005 lectures, and we'll go to, what was HIV lecture? Is that 24? Nope. 23? Well, we're looking here, folks. Hit the thumbs up button or hit the like button. Maybe we can attract some people to teach them the proper information about science. Yeah. Oh, that would be in the antiviral lecture, right? I think. Yeah, it's in the antiviral. So let's open antiviral lecture. I love having this resource at hand, my lectures, right? So the antivirals, I don't know, 21. Come on, computer. No, evolution, that's not it. Should be 19 then, maybe. I do have the numbers for you. It's okay. Grab a tea and just sit back. Vaccines, nope. Antivirals should be right after vaccines. That should be 20. Here we go, antivirals. So let's look at PrEP. Okay, PrEP reduces the risk of sexual transmission by over 90%. And it reduces the risk of transmission by intravenous drug use by over 70%. So not as good for IV drugs, but sex. And then if you couple it with a condom, you're close to 100%. Okay? Those are the numbers for PrEP. People are at high risk. Ubalubi, I wish I didn't have to drive to get around, specifically looking for postdocs in big cities so I can utilize beautiful public transport. I totally agree with you. As a grad student, I just walked to the lab and I loved it. I wish I could do that again. Hello, Visto. Welcome. 40 or 50 in Sydney. The thermometer has melted. Yeah, I heard the Australian Open is brutal. Now that I say that, I guess Little Rock is a big city in Arkansas. No good metro. Sad. It's all the fault of the oil companies who lobbied against funding of public transport in the U.S. Hello, Brian. Warmed up to minus 14. Oh my gosh, in Montreal. Tona, I found a new YouTube channel by the name of an immunologist who goes by LIL Scientist. Her name is Melanie Mathieu. Anybody else know her? I don't know her. Yes, TB is quite infectious and very difficult to treat. Yeah. Oh, Bureau of Atomic Scientists Doomsday Clock. I see. 12 seconds to midnight and nobody cares. Yeah. Ringo Starr got TB, was saved by antibiotics. 85 seconds to midnight on the Doomsday Clock. Yes, some phage therapy for TB, a different kind of TB. So there's Mycobacterium tuberculosis, which is the big killer. Then there's Mycobacterium smegmatis, which infects immunocompromised people, and phage therapy has been used to control that. We've done some of those papers. I forgot where, Twivo. Thank you for your contribution to MicrobeTV, John. Really appreciate your support of science programming. Hello, Jill. Welcome from Wilmington 20F. Wobble is great. Degenerate code makes me laugh. You got degenerates left and right, Mark. To Veronica, wobble is a term for astrophysics. Yeah, wobble is Alan's snow shovel, right? Yeah, Tona wants to know if this is the worst political times you have lived through in America. Well, Vietnam years were horrible. It was also lying, but people were accountable, right? That was pretty bad, but I think this is worse. I mean, it's hard because a lot of Americans died in Vietnam, right? So that's really bad. And I think that was unacceptable for, a manufactured war. But these have long-lasting consequences, right? We got out. The whole story was revealed. But what's happening now are going to screw up the U.S. for a long period of time. I can't imagine it being close to home being worse than it is now. Yeah. Daniel, we have an exam on virus structure. Can we go over that? Yeah, sure. Ask away. JJ wants to have can have a serious. He's asking. They're asking. Can having a serious measles infection in childhood have a long-term effect on T cells? I'm not aware of long-term effects on T cells. Just, the virus does reproduce in lymphocytes, so it will erase T and B cell memory for a time. But I'm not aware of long-term. But let's look it up, right? Does measles infection have long-term effect on T cells? Well, 2 to 3 years, immune amnesia, but not, 30, 40, 50 years, not that I'm aware of. By the way, when I used to write science fiction, the publisher said Marco Martin sounded like a pseudonym. It does, I guess. It does. Yesterday was Holocaust Remembrance Day, and I've been watching testimonial visitors from survivors. So Miss Blue Ocean, I called you by your other name. I'm sorry to do that. I didn't know you didn't want it said. TB and typhoid were rampant in the concentration camps. Those survivors were the strongest people. Fun question of the day. How long should we microwave takeout pizza, other takeout foods to kill Noro, COVID, flu, sneezed onto our food by food handlers? Oh, gosh. Oh, my gosh. Oh, my gosh. I would throw it out if someone sneezed on it. Come on. Go get a new one. Send me the bill. How long to microwave norovirus, because that's the toughest, contaminated food? I would just say throw it out. Should be thrown out. Even frickin' AI says it should be thrown out, not microwave. Noroviruses are notoriously difficult to kill. Some are resistant to 60 Celsius, which is higher than typical cooking. Microwaving does not provide a reliable internal temperature needed to inactivate virus throughout the food. So it doesn't give you a uniform internal temperature. So there you go, case. Throw it out. Uba Luby writes, 85 seconds to midnight is a changing measure of how close humanity is to nuclear war, climate change, biotechnology misuse. Our current trajectory is unsustainable. Scott, is it me or does Matters Microbial need some love? Yes, it does need love. The audio version is doing better than the video version. Yes. Lip sync with audio is fine at my end. Okay. Yeah, TB can remain dormant for decades. Hello, Cal. Call me Ishmael. Sorry. -12 in Maryland. Okay. The delay fixed it. That's good. Microwaving. The Nipah virus news says hundreds infected. No, 2 people were infected, and they checked 160 contacts, and nobody was positive. So that's wrong. Too many Americans are blinded by the cult of freedom and ignore the social contract. Patricia wants to know what happened with Amy. And Amy left the FDA. She's now at an institute in Philadelphia. So she's good. She's having fun. Excuse me. wondering if being vaccinated against COVID helps reduce common cold coronavirus symptoms. Not clear. There's some anecdotal evidence. I mean, there are cross-reactive antibody and T-cell responses. So it's possible, but I don't think we know for sure. Hello, Martha, in Charleston, South Carolina, where Michael Schmidt is from. Hello, Dragon, watching from Japan. Wonderful. Ohio. I think it's Ohio there. Ohio gozaimasu. I mean, gozaimasu. Gozaimasu. Ohio gozaimasu. Okay. How is it over there? Everything cool? Peter says, P-Class is a chap in New Jersey who normally does podcasts and YouTube content on electric vehicles. His video on his robot snowblower has gone viral, and he has many interviews on U.S. Telly. He said it as the storm began. Oh, that's cool. I like this robot snowblower. I actually went out Sunday morning and I have a snowblower that I haven't used in 2 years. I got tired of doing it, so I hired someone to do it. But there was so much snow in the morning, I said I might as well go out and do this. So the darn thing, of course, it's had gas in it for 2 years, which is the worst thing that you can do, right? And it wouldn't, and I'm pulling it, it wouldn't start. Then I see there's this thing where you could plug it into the wall. So I never done that. So I plugged it in and boom, it started right away. It's great. So it's amazing. You can, it's not recommended. You're supposed to drain the gas after every season, but it worked. And it was very, very light snow, so it was very easy to plow. But it was windy and it was blowing in my face. Up to code. Danny, immune systems have evolved over millions of years. Your best guess, how were Neanderthal immune systems different? I don't think they were very different. I think, we have some Neanderthal genes. About 3 to 4% of our genome is Neanderthal, which I think includes some immune genes. I think they're very similar, and they're just single nucleotide polymorphisms that distinguish them. Yeah. By the way, the podcast quality, video, audio graphics of TWIV, etc., is dramatically better than it was. Your small contributions helped make that happen. Well, actually, everyone's contributions help because I hire people to help me. I have audio and video editors now. I don't edit any of the podcasts anymore. What I am responsible for is all the verbiage, except for Mark Martin for Matters Microbial. I write all the summaries and I make the thumbnails. But the short videos, that's done by Chris Condayan, a video editor. The broadcast quality is pretty much broadcast TV. Well, thank you. I appreciate it very much. I always wanted to be technically proficient. I spent time to learn and spent money. So I'm glad it's looking good. Is there a phage universal to mycobacteria, or do you need one for each species? No, there's no universal one, and that can, as you treat an infection, the bacteria can change, and so then you need to redesign the phage, right? Select for a new one. I don't know about phage therapy for M. avium. I wouldn't be surprised, right? Let's look it up. Phage therapy for M. avium. Yeah, there are, and it's being worked on because there are drug-resistant M. avium. Yep. There you go. Mark Fuccio, nice to see you. First time in months I've been able to join office hours live. Abdul Rahman writes, Zofluza has been good for my patients so far. Very good. I'm glad you like it. What makes these times worse than Vietnam is social media. Yeah, you can lie a lot more on social media, can't you? For me, social media has enabled what we do at MicrobeTV, right? Because I consider YouTube and podcasts part of social media. So it is a tough situation, right? Never getting pizza again. Well, you can get pizza, but don't let people sneeze on them, right? Why would you do that? Well, it could be an accident, right? And Bacillus cereus can create stable toxins that will not cook out of food. So don't think you can microwave food to decontaminate it. Just throw it out. Visto writes, I suspect norovirus will survive nuclear warheads. It's a cruel world. Miss Blue Ocean says, I have not gotten sick this year. I went to Sarasota and back and wore my N95 in the airport and the plane. I have not eaten out or done much socializing because I was so sick last winter with Noro. Well, certainly masking and then not going out much, because in a restaurant it's hard, right? Because you have to eat, so you can't be putting your mask on and off. Take risks. How effective is coughing, sneezing in your elbow for preventing the spread of viruses all over the place? I'm not sure that a study has been done, right? How could you do that study? But the CDC recommends it. The old CDC recommended it, right? How effective is coughing into your elbow? Did you know it's called a vampire cough? Oh, yeah, that's right. I want your blood. Vampire cough. There's no scientific studies proving it stops all pathogens. However, it's better than, right? Just hawking outwards. No, it's got to be better. So I do that all the time. Do a good Dracula cough. Yeah. But it's not going to be perfect, no. But it's going to be better than just coughing out loud, right? Stephen asks, when it comes to measles, why weren't people able to eradicate it? What's the biggest challenge? Well, here's the problem. Many countries were not vaccinating. There were still a few 100,000 cases of measles a year in many countries because they can't vaccinate. You know, smallpox elimination was coordinated by WHO. They did it. And they're not coordinating a measles eradication. They're still working on polio, which they're never going to get. But anyway, so it's all about vaccinating. If you could vaccinate everyone, Stephen, you could eradicate measles, in theory. In theory. Are there other coronaviruses we can catch from our pets? I don't believe that you can get corona. There are plenty that infect pets, right? Let's see. No, there's not really any spread of, there's not really any spread of dog and cat coronaviruses to people. Yeah, here's a good quote by Isaac Asimov. There was a cult of ignorance in the U.S., and there always has been. It's nurtured by the false notion that democracy means that my ignorance is just as good as your knowledge. That's true. Yeah, it's I can't take it when people start to preach to me about viruses and they know nothing about them. Hello, MLE Lagunas. I'm staying warm. I really dress up. In fact, just between you guys, I'm wearing long underwear these days, and it is so great because usually I wear cotton jeans all winter, which is not so great, right? I don't have any wool pants to wear, but they're kind of cold, and the last couple of days I wear long underwear. Really nice. And very, very, very cozy and warm. Yeah. Somebody on Reddit said they used a leaf blower for the initial powdery snow. Yeah, it's very good. Case has a solution to stop chaos in the U.S. Demand all politicians running for office take a SPECT brain functional MRI scan before we hand over the nuke codes. I don't know if it's good enough. Visto says, how much do we inherit viral immunity from our parents? Do we inherit any long-term immunity? Well, I guess you mean antibodies and T cells, right? You get antibodies from your mother, but they don't last more than 6 months. But of course, you get all the genes. You get genes from your mother and father, right? For the genes of the immune system. And so that can have a long-term effect for sure. But no, in terms of the effectors, aside from the placental transfer, you don't get anything. No. I'm glad you like the punny TWIV titles. There's always so Alan likes to make punny TWIV titles. I like to be more straightforward. So that's where you get something like twin and, well, Tweevo, Nels likes to make the titles too. But I'm always fighting for straightforward titles so people can say, oh, hepatitis B, oh, cytomegalovirus, oh, herpes virus, dementia, right? So they can get attracted. And when you have a good informative title, we get more views. There's no question about it. Now, the audio only, that's a fixed audience, right? So I got, 50,000 subscribers to TWIV, and they all download it, right? And it doesn't grow very much. But the video is fickle. I have 140,000 subscribers, and some videos get watched 400 times, and some get watched 20,000 times, but never 140,000 times, right? So it's all about choice, because the audio podcast is automatically downloaded when you turn on your pod, so it's different. John writes, hardware and convenience stores sell stabilizer for fuel, so you can go a year or two with the gas going stale. I was told it wasn't going to work, but it did work. Nipah virus news from Tormento Oscuro. That's a nice name. Yeah, I already talked about it up at the top. I'm sorry if you missed it. Mycobacterium smegmatis was at first isolated on the spaceship Red Dwarf. No. Most of them cause skin infections, although lung infection and disseminated disease in immunocompromised people has been reported. Dragon writes, can you make a short from your last TWIV about the German response to Maha? Yeah, that's really good, right? I will ask Chris to make a short. I could make a short myself. What do I need to ask anyone? It's easy enough to do. It was really good, wasn't it? Yeah. Spamlet writes, I've had all the COVID and flu shots. I still have a runny nose, though I rarely go out or have visitors. Well, there are lots of other things that can cause runny nose, right? But you rarely go out. So you go out now and then. You have to at some point, and there you go. Lori writes that 3% of our DNA that is Neanderthal. Does everyone have more or less the same 3%? More or less the same. Yeah, more or less the same. And make another short with respect to your response to the news article Amy relayed to you. Yeah, that's another good one, too. Can hospital sewage be a good phage bank? I think so. I'm not sure people use it, though. People tend to use soil. But yeah, that would be good. Let's look up sources of phages for therapy. Of course, many places have huge collections of phages. Environmental samples, clinical waste, there you go, hospital waste, and biobanks. Sewage, water, wastewater, rivers, lakes, soil, the gut, the gut, mouth, urine. Yeah. Places that have high bacterial density, basically, you could look for phages. Yeah. Tona writes, imagine Amy's paycheck from her former job being signed by the big orange yuck man. Is that right? Do you think it would be signed by... But I think it's direct deposit. You don't see a check anymore, right? Yeah, Mark, I didn't read it today, but I read it on Twiv. The criticism of Germany and the rebuke. Yeah, that was really good. Here's a possible, Nevitz writes, here's a possible German response to this administration. Back, Pfeifengeschicht, a unique term for a person who's obnoxious, smug, or annoying. Face seems to beg for a slap or punch. Back Pfeifengeschicht. Wow, that's cool. Pretty sure we cough into our elbows, so we don't go touching things with our freshly coughed into hands. Yeah, that's another thing. If you do this, so some people just spew into the air, you know, which is bad. Okay, I want to do some newsy thingy before I go any further. Let's see here. I thought I had queued it up, but obviously I went away. I want to go over this. I have to take Jill away here. Away. This is Beyond the Noise by Paul Offit. We're going to record this on Monday, but I wanted to tell you about it because it's so frightening. This guy is just out of his mind. And you got to understand that most people in this administration are similar. So Dr. Kirk Milhoan is the new chair of the ASIP, and he was on a podcast called Why Should I Trust You? So here's the interview. Do you think that vaccines are appropriately tested for study? Let me blow this up here. Milhoan, no, they haven't been. They've only been studied for efficacy. And Paul Offit writes, what is he talking about? Of course they've been tested for study. They haven't been studied just for efficacy. So, as Paul said, before licensure, vaccines are tested in prospective, placebo-controlled trials involving tens of thousands of subjects to determine both safety and efficacy. And then after that, you give it to millions of people and you continue to follow. And that's how myocarditis was picked up and clotting was picked up in the COVID vaccines. So he's just wrong. He's lying. He should know that. So Paul said, the interviewer asked another question, and he said, I think as you look at polio, we need not to be afraid to consider that we are in a different time now than we were then. Our sanitation is different. Our risk of disease is different. No, you don't know what you're talking about, Milhoan. The sanitation is what made polio an epidemic disease. Why don't you know what you're talking about? Gosh, when you have an authority, a supposed authority who doesn't know anything. Let me explain what this is about, okay? Well, now we have to cut out Jill. So up until early 1900s, there was no sanitation anywhere. Every baby was born and got poliovirus infection immediately. But guess what? It had its mother's antibodies, and it was protected from paralysis. So then, because polio was circulating, because sewage is everywhere. But then, 1900, we developed the toilet. According to Dixon, the greatest invention of humanity, the toilet and sewers. We now take all the feces away from the streets. Kids don't get infected anymore in their first few months of life with poliovirus. Now they get infected later when they don't have their mother's antibodies anymore. And boom, you have outbreaks of thousands of cases of polio. So Dr. Milhoan, it doesn't matter that we have great sanitation. Actually, you don't get it now from sanitation. You get it from other people, because we don't have sewers in the street. Bozo. Oh, my God. Here, next one. Milhoan, many of the risks of measles without having a vaccine was in the 1960s. We take care of children much differently now. Our ability to have pediatric hospitals, children's hospitals, pediatric ICUs. What we're going to have now is a real-world experience of when unvaccinated people get measles. What is the new incidence of hospitalization? What's the incidence of death? Oh, my God, this is another idiot. Well, it's the same guy. And Paul writes, the first measles vaccine was available in 1963. Before that, every year, about 48,000 children would be hospitalized and 500 would die. Hospitalization is caused by pneumonia or dehydration. Our ability to take care of pneumonia with oxygen or ventilation or dehydration with IV has not changed since that time. So he's BSing you, saying we take care of children much differently now. The death rate from measles in 1963, 1 to 3 per 1,000 cases. The past year in the U.S., 3 people have died among the roughly 2,000 reported cases. No different than the mortality rate 60 years ago. And Paul writes, again, what is Milhon talking about? Okay, so then the interviewer says, where do you see the line for individual autonomy versus you are infringing on my child's safety by the choice you are making? Milhon, what if a child gets a measles vaccine to protect your immunocompromised child and gets a negative consequence from that? Wasn't that your child causing that child to be harmed? Milhon, and this is Paul writing, is arguing that any parent could reasonably decide not to get a measles vaccine because they believe the risks outweigh the benefits. Measles virus causes pneumonia, dehydration, encephalitis that can result in blindness and deafness. Measles vaccine doesn't cause any of these problems. So why would any parent reasonably choose not to get the vaccine? Okay, Milhon, again, it's been very important to us, the members of the committee, that what we were doing is returning individual autonomy to the first order, not public health, but individual autonomy to the first order. This is just nonsense. And there it is, writes Paul. Individual freedom trumps public health. That's why, according to Milhon, the individual has a right to make poorly informed decisions that put not only their child but other children at risk. Bioethicist Art Kaplan has written, the likely increases in childhood illness and death appear to be worth the price of freedom to choose whether to vaccinate. It's not just bad policy, it's bad ethics. Okay, back to the interviewer. You're probably going to be getting a lot of reports and a lot of files and a lot of data that's been established, things that we've looked at and pursued and investigated. Milhon, that's not science. That's not necessarily science. Science is what I observe. And Paul Offit writes, I have no idea what this means. Well, Milhon is saying that he's the science. He can decide what's right and what's wrong. He doesn't need any stinking data. Just like RFK Jr. says, I don't need experts. And Milhon writes, if you allow flexibility on certain vaccines, you actually then enhance uptake in other ones that are crucial, like MMR and polio and things like that. No, vaccine rates are eroding in the U.S. We have measles outbreak. We have more flu in children than ever. Cases of tetanus, whooping cough. He's saying if we loosen vaccine recommendations, then vaccine rates will increase. On what planet does he imagine this happening? Oh, and here's a good one. Milhon, I've never seen a case of congenital rubella affecting a child in my career. Of course you haven't, moron, because the vaccine has eliminated rubella from the U.S. Before the rubella vaccine, about 20,000 children in the U.S. whose mothers were infected with rubella were delivered children who were blind or deaf or had heart defects. 20,000 children. The vaccine eliminated rubella from the U.S. By 2005, but it's still common in other countries, and there's still a lot of congenital rubella syndrome. And listen to this at the end of the podcast. The host, Brinda Adhikari, made a final plea. I also ask that you hold space for these moms who are really worried about infectious disease and are really worried about them making a comeback should uptake of these vaccines go crashing down. To hold space for all of that, because I think that's what I am entrusting in my government, to not hurt me and look out for me in some way. So I just ask you, hold space for those voices. Will you do that for me? Can you imagine? She needed to ask this idiot to hold space for people who want to be protected. This is a contrarian view that should not be in power. Vaccines help. They help a lot. They prevent infectious disease. They prevent suffering and death. Not taking them is not an option. Public health doesn't work if you get the option. I have no problem taking vaccines. Absolutely none. I know them really well, and believe me, in most people, they are fine. Oh my gosh. Isn't that horrible? Isn't that horrible? Oh, yeah. Let's go back to you folks. Since bird flu virus latches onto certain sialic acids on certain tissues, can viruses be used to identify markers of differentiation? Well, there are better ways to do that. We don't need to use viruses to do that. Why does measles, Peter asks, have a high reproduction rate? So, a couple of reasons. The amount of virus, so that you make a lot of virus in your respiratory tract, more than most other viruses, so a higher virus titer. So there's a lot more virus coming out to infect other people. And you don't need as much virus to start an infection. And so the virus that you need to start an infection can fit into a tiny droplet that you expel that can travel a long way and infect more people. I think those are some of the reasons. Also, it's immunosuppressive, and that helps as well. And Marco writes, you were lucky. I needed to drain my old gas out of the carburetor bowl this year. Need to take part of the carburetor apart. Oh, my God. It was freezing. Blowing wind and freezing. I wasn't about to do that. No. I guess I could have gone in the garage, but. Is not environment super important? Yes, of course. But measles seems to spread in many environments, right? What do I know about the virus that affects oranges in Florida? Citrus tristeza virus. Let's look it up. Let's make sure. It's citrus tristeza virus. Yeah. So we actually had a lady from Florida who researches this virus. So it is it's a rod-like virus with an RNA genome, and it's a very large virus. It infects several species of the genus Citrus, including oranges of all kinds, limes, grapefruit, and mandarin, and also other plants as well. And it's a big problem in many places, including Florida, where it is really messing up the plants. It's the economically most important and most damaging virus of citrus trees. It spreads quickly. It can do damage in many ways. Has killed more than 80 million trees worldwide, mainly in South Africa and the U.S. and Brazil and Argentina. It's hard to stop it from spreading. That's part of the problem. Citrus tristeza. UO20. How do manufacturers update the COVID and flu vaccines every year? They monitor viruses in patients. For flu, there are many, many laboratories worldwide that collect samples from patients and determine how much it's changed and whether we should update the vaccine, how it's changed with respect to vaccine-induced immunity. And a similar thing for COVID, although not as extensive a network globally. Is the spike protein causing scarring on heart tissue? The SARS-CoV-2 spike protein. I don't know. Let's look it up. So basically, the spike does harm heart tissue, mainly by inflammation, right? Also by blood clots, micro blood clots, and so forth. So, so basically, infection increases your risk of a lot of heart problems, like heart failure, arrhythmias, heart attacks, even in healthy people. And so that's the virus. The vaccine does not do that. It causes myocarditis in a small fraction of young men mainly, but the vaccine does not do that, and so that's why it's good to prevent infection. Did I see the paper on prototaxoides finding it isn't a fungus, but could have been a unique life form? That's interesting. It's interesting. We have to, I'll look at that. Yeah. Okay. Now I want to do a mini lecture before it gets too late. It's already 9:16. You know, time is flying. This one is a surprise. You don't know. You have no idea what I'm going to do. And this wasn't suggested by anyone, but it's about poliovirus receptor, which we talked about how my lab identified it a couple of weeks ago. But now I want to talk about the function. What does this protein do? Cell receptors for viruses have functions in the cell, right? Here's a whole slew of cell surface proteins and glycoproteins and sugars that are receptors for different viruses, and they all have functions in the cell. So, HIV, the CD4 molecule is an important molecule on CD4 T cells, and the co-receptor is a chemokine receptor. So when we first identified the poliovirus receptor, it was a new gene that anyone had found. Nobody had seen it before. So we wanted to know what it did. And we published this paper in 1992, Mary Morrison and myself, cloning an expression of a murine homologue of the human poliovirus receptor gene. So we thought, if there's a homologue in mice, then maybe we can study what this protein does. So we called it the murine poliovirus receptor homologue, or MPH. Okay, that's going to change. So here's the structure of the poliovirus receptor, also known as CD155. And in mice, there is a very similar protein with 3 domains, transmembrane protein, which we call MPH, and it shares 64, 68, and 80% similarity at the amino acid level in domains 1, 2, and 3. Okay? Now, domain 1 of PVR is where the virus binds. And so already you see 64% amino acid identity. Well, this receptor, MPH, is not going to be a polio receptor. And we already know that mice are generally not infected with polioviruses, although there are some exceptions. That's why we had to make transgenic mice with the poliovirus receptor gene to make a mouse model. But we asked the question, does MPH bind poliovirus? And we start with mouse L cells that don't have the poliovirus receptor. We put in DNA encoding the murine homologue, MPH, and then we infect them with poliovirus. Okay? Very straightforward assay. Here's what we get. All this has been published. So here's on the left the DNA that we put into the cells, and then we infect, and the PFU per mil of poliovirus at 0 and 48 hours after infection. So here are 3 controls. So first, herring sperm DNA. Why herring sperm? Because you can buy tons of it, okay? It's very available. It's like foreskins, all right? You can get lots of it. And so we put herring sperm DNA into the cells, and you get a 4 log increase, but it's not really replication. This is just virus that's stuck to the cells, and so it's input because there's no poliovirus receptor in these cells. So then we put the vector that we're going to put MPH in, and again, we don't get polio replication. But then if we put the poliovirus receptor, PVR, into these L cells, we see you get 10 to the 7th PFU, 7 logs of virus. So that's conferring susceptibility. Okay, so now we make a series of experiments. We have, first of all, this is, again, the structure of the poliovirus receptor with the hatched circles. That's poliovirus receptor. We put that in cells, and we get 7 logs of virus. Okay, so that makes sense that it's conferring susceptibility. Then we put MPH in. This is all MPH, right, shown by the different coloring. So it's the murine homologue. You don't get any susceptibility to poliovirus. So MPH is not a receptor for poliovirus. Then we make some domain swaps. We take MPH and we swap domain 1 with the domain 1 of poliovirus receptor. Look at that. Now the cells can bind and utilize this receptor to get into cells. So it's a chimeric receptor. We put domain 1 of poliovirus receptor onto domains 2 and 3 of MPH. That works. So clearly domain 1 is really important. And then we put domain 1 and 2 on because it turns out there's a slight difference with just domain 1, but it works as well. So domain 1 of MPH can be swapped and lead to being a receptor for polio. So then we got more granular. We published this paper 2 years later. Homolog scanning mutagenesis reveals poliovirus receptor residues important for virus binding and replication. Okay? So what is this homolog scanning mutagenesis? It means if you have two proteins that are orthologs, they're related to each other, you can swap amino acids. So we take poliovirus receptor amino acids and we can swap them from with the same amino acids from MPH or vice versa. Okay, to see at the amino acid level. I've shown you, domain 1, right? Domain 1 is important, but what about at the individual amino acids? So here we go. Again, we have, this is just to show you the names of these cells. So HeLa cells has a wild-type polio receptor. L cells has MPH. 20B is L cells with the poliovirus receptor gene. And then we have E2 and H52 with domains 1 or 2 of PVR onto MPH. And here are the growth curves for these. Let's look at MOI 10 PFU per cell. So all of these work well, with the exception of the triangle, which is L cells. So L cells have MPH, only poliovirus does not infect them, right? So we're looking at total virus produced with time. And you see all of these cells, HeLa, 20B, E2, H52, they all can produce poliovirus with time, but not L cells. But what's interesting is when you go way down in the MOI, 0.001 plaque-forming units per cell. So in other words, that's how much virus you add to the culture. And you see that there's some difference here. The circle, which is E2 only has domain 1 of PVR on MPH. That's not doing as well. So if you have H52, that's the squares, the open squares, that's doing as well as L cells or HeLa cells, L cells with PVR or HeLa cells. But only domain 1 is not as good as domains 1 and 2. So somehow domain 2 is contributing something. Okay? So that's pretty cool. Now, here's the sequence of domain 1 of MPH and PVR, the amino acid sequence. Here's the structure. So it's domain 1. It's got a lot of anti-parallel beta strands and making two sheets on two sides of the molecule. And you can see there are amino acid differences between MPH and PVR. And we've highlighted in asterisk the ones that we have swapped. We have swapped into PVR the MPH amino acid. So here's A. A is located whereas here in this loop we did a swap of the amino acids. We substituted QV for LP. Same thing with B, C, D, G, J, I, E, H, and F. So we made 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 swaps of single or multiple amino acids. And then we put these in. We put the DNA in cells and we infect with poliovirus and we see what happens. Okay, so here are the growth curves. And these are all at MOI of 10. So here are our HeLa cells and our PVR expressing mouse cells and our mouse cells as controls. So here we have the growth curve in B for the mutants A, E, and H. So A, E, H, where is it? Right there. Those swaps don't make any difference, as you can see. Neither do C, F, and H. J here doesn't make any difference. But look at this one here. D, G, G, and I all make a big difference. Here's D. Where's D? D, G, J. I'm sorry, I. The I is over here. These all make a big difference in replication. And the other changes don't matter. So the ability to bind poliovirus is located in these two beta strands here. And that's interesting because for HIV binding to CD4, it's the same. So CD4 is an immunoglobulin-like molecule, and domain 1 looks just like poliovirus domain 1 right here. And in fact, there is an amino acid at 43 of CD4. It's crucial for HIV binding. And if you make amino acid substitutions, you abolish interaction with GP120 of HIV. So here's position 43. It's a it's a phenylalanine. Okay? And if you change this in HIV, you abolish binding. Interestingly, the same way a single amino acid change at the homologous residue in PVR, which would be Q55F, also abolishes poliovirus binding. So here's Q55F. So that's mutant D. We change it to F from Q. That is mutant D. So let's see, that's in this place here where the same amino acid is present in CD4 and controls HIV binding. So let's look at the D mutation. And you can see here, D31-3, it's right there, completely abolishes poliovirus infection. And so it's just interesting that the two different viruses have similar binding sites on their receptors. Okay, but you may be saying, Racaniello, what about the function? That's what you set out to do. Okay, we got to it. Here's the paper we published, Defects in Nuclear and Cytoskeletal Morphology and Mitochondrial Localization in Sperm of Mice Lacking Nectin-2, a component of cell-cell adherens junctions. So now MPH has been renamed Nectin-2 by the namers that be. I didn't have anything to do with it. They just renamed it. But anyway, this is work by my student, Michael Bouchard, a PhD student. And what he did was to knock out the murine MPH gene, or Nectin-2. Okay? The way he did that was, here's a map of the MPH gene in mice. He designed a DNA that he would put into cells that would disrupt the gene, and he put this into embryonic stem cells and put them into pregnant mice, and the offspring were knocked out. So here's a southern blot showing you the wild type gene, and then the heterozygous knockouts. They have a smaller gene, a smaller DNA, and then the homozygous knockouts, both copies of the DNA are disrupted. Okay, so now we've knocked out the MPH gene in mice. The homozygous mutant mice have no overt developmental abnormalities, but when we cross them, we don't get progeny. In fact, the male Nectin-2 null mice are infertile. The female mice, so the male and female heterozygotes, and the female homozygous knockout mice are fertile, but the Nectin homozygous null male mice are infertile. And in fact, when we mate 25 Nectin null homozygous male mice with females, we see copulation plugs showing that mating had occurred, but no pregnancies were observed. So we've now become a lab interested in development in mice. And, we're a virology lab, but this was really interesting. So we thought we should study it. So these are the sperm counts and the testicular weights of the testes and the epididymis in male, wild type, and homozygous null mice. So wild type mice, here is the sperm count. It's the same in the nulls. The weight of the testes is the same. The epididymis is the same. So that doesn't explain the infertility. But where is this? Oh, where is that slide? Did I not put it in? Oh, how could that be? Well, I have to get this slide. It turns out that the sperm are morphologically aberrant, and I had put in a lovely picture here. Let me see if I can find it. Wait a second. Yeah, here it is. I should be able to put in a slide here. So, add slide, and we'll paste it in. No, you didn't like that, huh? Let's try it again. Oh, I have to show this to you because it's so beautiful. Okay, hang on, I'm almost there. Okay. This is the best part. I remember when he did this experiment, he was so excited to see this. There we go. So these are wild type sperm from wild type mice on the left, and sperm from homozygous null MPH knockout mice. You can see they're all messed up. The heads are aberrant, the tails are all bent and twisty, and they're not uniform thickness. I mean, the ones on the left are gorgeous, right? And the knockout sperm are all screwed up, and that's probably why the males are not fertile. If you stain the sperm with mitochondria stain, MitoTracker detects mitochondria. Here's a wild type sperm on the left. You see the mitochondria are all in the neck, which is what beats, right? They need energy. But look at the homozygous null mice. There's some mitochondria in the head and not much in the tails. So something is really wrong here. Now remember, we've knocked out the poliovirus receptor homolog gene. Then we did electron microscopy of the sperm. So here's a wild type sperm here, an A, beautiful head, full of DNA, right? And then the neck is full of mitochondria. Look, they're all lined up on either side of the actin that makes structural rigidity. And if you look at the sperm from null mice, look on the right, the head, this is the head. It is totally screwed up, and there are mitochondria in the head, as suggested by the staining I showed you. And if you look at the tail, you can see it's all messed up. The mitochondria all jumbled around here, and the actin in this one on the right D is all screwed up. So knocking out the PVR homolog in mice makes males make aberrant sperm. We made an antibody to Nectin-2, and we stained mouse seminiferous tubules with the antibody. You can see these cells are making Nectin-2, and these are sperm maturing in the tubules, but Nectin null mice, they don't make the protein. And here on the lower left is a knockout sperm head stained with Nectin-2, and there's very little expression, whereas in wild type sperm, Nectin-2 nicely stains the tail of the sperm. Right, but there was nothing in the knockout at all. And finally, we stained the sperm with actin because actin, remember, makes up a big component of the tail. And wild-type sperm stain nicely with actin, and there's very little actin in the knockout sperm. And so why are these sperm defective? Well, Nectin-2 is a component of cell anchoring junctions, which are called adherens junctions, and it's linked to actin through a binding protein called ephedrin. And these junctions connect the cytoskeletal elements of neighboring cells. They make a transcellular network that's structurally sound, and they also regulate shape and differentiation through signaling. So we think that nectin regulates directly or indirectly cytoskeletal structure during spermiogenesis. And here's a picture of how that can happen. So here we have F-actin, which makes up the structural part of the tail, and then nectin is coordinating interactions with this scaffold protein that binds actin and also other adhesion proteins as well. So you get rid of actin, you mess up the ability of actin to do its job. So is poliovirus receptor required for spermatogenesis in humans? So I was so excited by these findings. I wrote a grant saying this could be a target for a spermicide, right? And I never got the grant because it turns out that sperm development is an old boys club that you can't break into. And they said, oh, this virologist wants to work on sperm development. No, and I never got funded, so we never did it. But it turns out we did some preliminary experiment. In transgenic mice with the human polio receptor gene, the protein is expressed in the seminiferous epithelium in a pattern similar to what we observed for nectin-2 in mice. And furthermore, if we put PVR into nectin-2 null mice, it partially overcomes the morphological defects in sperm. So maybe the answer is yes. But so just to summarize, PVR has multiple functions in humans, and this is in addition to MPH in mice. It's a cell adhesion and junctional organization protein. It localizes to adherence junctions just like MPH. It binds a fade-in. It participates in cell-cell adhesion. So it's really important for epithelial sheet remodeling, wound healing, and tissue morphogenesis. But it also regulates cell migration and polarity by modulating other proteins. So it's important in development, tissue repair, and cancer invasion. And we now know it's an immune checkpoint ligand. It's a key ligand for receptors on NK cells and T cells. It's often upregulated in stressed, infected, or transformed cells. And the consequence is it acts as an immune rheostat, tipping towards immune suppression in tumors. It also plays a role in cancer biology. It's overexpressed in many cancers, and it correlates with increased invasiveness, poor prognosis, and immune evasion. So really important in cancer biology as well. So who knew any of this when I started? I just wanted to know what the function of the polio receptor is. In these last few slides, other people have done this work. But this is why scientists are curious, and it illustrates the role of serendipity, that you can't predict where things are going to go. Okay. Hope you like that, because nobody really pays any attention to this stuff. And I thought I would tell it one last time. Okay, let's go back to the comments here before we read our poem. Visto writes, so it is that mother's antibodies, although not lasting, let a child survive long enough for their own immune system to learn its own response. That's right. But remember, you're dependent on your mother's infectious experience, right? And that's why if the mother is vaccinated properly, then the child will inherit those antibodies. And so now we have a respiratory syncytial virus vaccine for pregnant mothers. The antibodies she makes are passed on to protect the children. Mark writes, my father, who attended Columbia and then Columbia Law, had a German friend. As teenagers, he taught us a German phrase that applies to RFK Jr. Dumm geboren und nicht dazugelernt. Translation, born dumb and learned nothing since then. That's great. I love it. Born dumb and learned nothing since then. So Kay said, asked about transparency in pharma, and Nevitz writes, I worked for a company that manufactured vaccines for 10 years. It was top to bottom FDA rules through every stage of the trials and manufacturing. It was drilled into us. Thank you very much for that. What's the rate of cerebral measles recurrence in adolescent males? You're talking about subacute sclerosing panencephalitis? You want to know the rate of that? Okay, let's look it up. I don't know it offhand. SSPE rate. 4 to 11 cases per 100,000 measles cases. If someone with norovirus pukes near you, you get noro. Yes, especially on an airplane, right? Yes, yes. Hello, Tractor Poodle from Bangkok, 26C. Oh my gosh, send some of that warmth over here. TB writes, passive immunization via breastfeeding. Is by polio possible if mother is newly, freshly infected, i.e. washing cotton diapers? So are you asking if polio can be transmitted by breastfeeding? I don't believe it can. Because it doesn't reproduce in the breast. Yeah, it doesn't really, it's not transmitted via breastfeeding. Yes, Sandy, this guy is a gigantic asshole. How do they find the worst? Everybody in the administration is basically an asshole, and they think they're the best. But according to GOP, there is no bodily autonomy for women. The conservatives on SCOTUS unconstitutionally repealed Roe that recognized a woman's right to end a pregnancy. Yes, and viruses have reproductive rights, but not women. In Idaho, Rita says the child vaccination rate is at 75%. Now we have measles in some schools. Polka Dot Pup read Bad Faith. Yes, it's a very good book by Paul Offit. Will keeps a list of expert idiots. Milhone is number 73. Yep. Laura writes, you should read up about PFAS building up in the brain over time. It causes epinephrine to build up like mercury. Mercury is known for Mad Hatters. They act like mad Trumpers. Vanity got a Tdap on Saturday. I didn't feel anything, but my Oura ring recognized my immune system was engaged. So what does the ring tell you? You have a low-grade fever, or what can it detect that says you're having inflammation? Tess Siegel writes, the lab leak people say the furin cleavage site is supercharged and therefore could not have naturally evolved. It is not supercharged. They don't know what they're talking about. Of course it could. There's plenty of evidence that it's naturally evolved. They don't know what they're saying. They're lying. You know, I've gone over this in previous TWIVs. It is just not. If you want me to talk about the furin cleavage site in a mini lecture, I'd be happy to do that. Let me know if you want it. But no, the lab leak people don't know what they're talking about. I don't care who they are. They know not what they say. Starman, the damage from long COVID is multifactorial. But, I mean, cell death is part of it, but inflammation is a big part of it as well. And you need to have infection for it to happen. But it's certainly a consequence of inflammation. But why it happens in certain people, that's the key. We just don't know. We just don't know. Yeah, so the work I told you today was 1992. There was no complete genome, so we had to clone our own genes and map them for sure. Yeah. Laura, a scientist friend of mine, thinks that PFAS is going to get all of us before warming. Overshoot has a lot to do with it. We have definitely overshot our planetary boundaries. Yes, an IMI of 10 is around one burst of virus reproduction. Nevitz writes, this is a beautiful example where research from one protein structure, CD4 protein HIV infection, can inform research on another virus. This always happens in biological science. Yep. Elizabeth, science, go in for the viruses, stay for the mouse sperm count experiments. Yes. Hello, Ruby from Machiasport, Maine 3F. Listening on 2X to catch up. All right, I'm glad you liked it. Why did the poliovirus pick on CD155? Random stuff, right? Viruses evolve. A few viruses are made that can bind CD155, and boom, there you have it. That's right. Penicillin was an accidental find, too. There are lots of such findings, yeah. Helen, thank you so much for this. The developmental biologist that I am is very happy right now. And yes, repro people form a very close group. Hello, Dorothy in Duluth. It has warmed up to 0F. In your figure 1, why did your number of virions at time 0 so much lower for 2 of your samples? Let's go back and look. It sounds like a good question. All right, figure 1. Why did your number of virions at time 0 go? Oh, so that's a growth curve. Okay, that's a good question. So what's happening here is that all these cells have poliovirus receptor on them. So they're binding virus. And so that's what you see is the virus bound, whereas the mouse cells don't have any poliovirus receptor. So the only binding, this 10 to the 4 background, is background nonspecific binding. Okay, that's what that is. Yeah. Okay. Is the human polio receptor unable to mutate away from susceptibility because of its role in reproduction? I think you're right. I think that it cannot change to avoid poliovirus because it has so many of these essential functions. Yeah. Good question. The Oura ring detects changes in body temperature and heart rate variations, among other things. I've learned a lot of insults for stupid people from this episode. Cool. About the Oura ring. A fun poem you might like to read is Bad Sir Brian, Botany by A. Milne. Okay, I'll put it on my list. Here's a measles risk level tracker by zip code from Vanity. Thank you very much. Tona writes, my son's watch, which senses his biorhythms, sensed that he had what turned out to be COVID before the test kit even said he had it. That's interesting. That's interesting. Yeah, it's too bad we need a measles tracker in 2026 in the U.S. Okay, let's read a couple of poems, okay? So my poet for tonight is William Carlos Williams. He was born in Rutherford, New Jersey. He was not only a poet, but a physician. He practiced pediatrics and general medicine and was affiliated with Passaic General Hospital, and he was the hospital's chief of pediatrics from 1924 until he died in 1963. And the hospital is now known as St. Mary's General Hospital, has paid tribute to him with a plaque that states, "We walk the wards that Williams walked." And my dad used to work at this hospital, Passaic General, and then St. Mary's, and so he knew him and also his son. So I have 3 short poems by William Carlos Williams. The first is The Fool's Song. I tried to put a bird in a cage. Oh, fool that I am, for the bird was truth. Sing merrily, truth. I tried to put truth in a cage. And when I had the bird in the cage, oh, fool that I am, why, it broke my pretty cage. Sing merrily, truth. I tried to put truth in a cage. And when the bird was flown from the cage, oh, fool that I am, why, I had nor bird nor cage. Sing merrily, truth. I tried to put truth in a cage. Hi-ho, truth in a cage. I think that's really prescient for these days. Then we have one called This Is Just to Say. This is one of his most popular ones. I have eaten the plums that were in the icebox and which you were probably saving for breakfast. Forgive me, they were delicious, so sweet and so cold. I'm going to read it again because it's so cool. I have eaten the plums that were in the icebox and which you were probably saving for breakfast. Forgive me, they were delicious, so sweet and so cold. So it's called This Is Just to Say. And finally, perhaps his most famous poem, The Red Wheelbarrow. So much depends upon a red wheelbarrow glazed with rainwater beside the white chickens. I mean, it's beautiful. I have no idea what it means, but it's really beautiful. I like it a lot. So William Carlos Williams, really nice American poet. Yes, Dorothy, Tigande is dangerous to infants exposed if their mother has an active case when the baby is born. You can have, it can get into the baby's brain and cause horrible things. Yes, you cannot clean cat boxes when you're pregnant. Absolutely right. Okay, folks, that'll do it for another Office Hours. I want to thank all of you for coming. I hope you all hit the like button before you go tonight. I want to thank our moderators for being here. We had Vanity Nutrition. We had Les Fabi. And we had Tom Steinberg. Thank you so much for moderating. I really appreciate it. And all of you who came and participated and asked great questions. Really appreciate your coming. I will be back next week. Let's see. Today is the 28th, and next Wednesday is the 4th of February. So that'll be it for January, right? And before the fastest 2 hours a week. Thanks me. Thank you so much. And here's one from Nevitz. The fog comes on little cat feet. It sits looking over harbor and city on silent haunches and then moves on. Lovely. Carl Sandburg. Yeah, Sandburg is another one we could do. Good night, everyone. Have a good week. Be safe. Stay warm. Don't fall. See you next time. Bye-bye.